Take a look at your syllabus quickly, if you would. We have lecture or lab exam is this week. About 1/3 of the class has already survived that exam. Some of you will be taking it tonight. Some of you will be taking it on Thursday. Your lab reports, your final draft is due this week in lab. Remember to bring 2 copies of your papers, and then finally your case studies are due by Thursday. So bring those. If you're in hybrid, bring those to lecture or to lab. If you're here, bring them to lecture anytime between now and Thursday. Remember that for the case study, you want to make sure you upload your answers to SafeAssign, as well as turn in a hard copy with your signed "We Played Well Together" form in the front of that before I'll grade that. The second case studies are available and open to you. If all went well in your group, you can just continue to do a second case study as the same group. If your group has fragmented or you've lost a member of your group and you need another kind of reestablish a new group, let me know, and I'll try to put you guys together in a group so you can finish the second case study more efficiently. The fourth lecture exam is going to cover lymphatic, digestive, and respiratory. I sent out a message last week. I hope it made sense to you. As you can imagine, I have the same exact number of instructional minutes in the summer as I do in the fall. That's why the lectures go from 1 hour and 25 minutes to 2 hours and 10 minutes. And the problem is I give you 2 hours and 10 minutes to do a lecture exam. So I lose 40 minutes almost every single lecture in instructional time. And so that over the semester drives me to have to put a couple of lectures online. So this summer, I've given you a lymphatic presentation. It's on one YouTube. It's actually on 2 YouTube links. They're there for you. That is completely and totally on YouTube, and I've given you that for some reason. Those pages got omitted from your supplement. So I've given you a copy of those. If you need to get a copy of those, I'll have them for you at lab tonight or on Thursday as well. I'll start today with digestive system. We'll go through this today, and probably it'll hang over into Thursday. But when's our exam? Next Thursday. So actually, I really need to get through digestive system mostly today and a little bit on Thursday. And then respiratory system will take up the rest of our time on Thursday and next Tuesday. Your lab this week introduces the respiratory system. The lab next week will go deeper into the respiratory system. So it'll all come together as it has in the past on the exam. Then our last and final lecture exam, just looking ahead, will be Thursday of the first week of August. And that will be a full-length exam. It'll be a little bit of cumulative. That's going to cover urinary and reproductive and a little bit on acid-base regulation. There will be a total of 12 quizzes throughout the semester, including urinary and reproductive quizzes. I will drop the lowest 2 quizzes. There will also be 2 totally additional extra credit quizzes that I think one should be available now, and if it isn't, it will be very soon. It says extra credit quiz 1. It's there. It's on a topic we have not discussed. So it is an open book, kind of exploratory. One is on energetics, glycolysis, and that kind of thing. The other one later on will be on human development. So there are 2 topics that there just wasn't time to include, but are interesting in the scope of what we're doing. Those are open book, open resource. Again, you can come and go to those quizzes. Do 2 or 3 questions every day. Don't stress out about it. Do the very best you can. Those will be extra points in addition to everything else. You get your 2 case studies, you have your mastering points, you have your lab report. So there are a number of things coming your way that will help your grade. Any questions logistically from me before we get started with the digestive system?
I had a question on the lymphatic system.
Lymphatic system.
Yes, when they say that when cancer gets into your lymphatic system, that it can spread to the rest of your body. Now,
yes.
Does it spread through the blood then? Because
cancer,
once it gets into the lymphatics, I thought stuff couldn't get back out because of the overlapping.
No, lymphatic, once cancer gets in, cancer spreads through blood but especially through lymphatics. So it definitely does spread, and things will spread from lymph node to lymph node, which is why we do testing on lymph nodes, and the cancer can be there. And it also can escape out of those structures. Remember, all the lymphatic fluid does drain back into the circulatory system. And so all of your lymphatic fluid does drain back into and become part of your plasma of your blood. And so there is an ability for cells to just take off and go wherever they want to. Yeah. Okay, let's take a look at the digestive system. There's a number of sections that are purely anatomical review. I've got them here as review. I'm going to skip over those and have you look at those. So 1, 2, 5, 10, 13, 16, 18. Those are just anatomical sections of review, parts of the stomach, parts of the intestine, that kind of thing. So but I did include, as I always do, I did include those slides in the packet. Now let's just start off though, let's think about this whole idea. The digestive system is providing nutrients for all of the 75 trillion cells in your body, right? All the energy comes in and is required for the growth of the cells. And the digestive system is working with other systems, as all systems do, that are working. So the digestive system is working with the cardiovascular system. Clearly, it takes the pumping of the heart to transport these nutrients. It takes the respiratory system to transport the oxygen that's also necessary for energetics, and the urinary system, which is constantly helping to flush out acids that are created by your body. Now, even when your body takes in glucose, and we'll talk more about this when we get to the urinary system and the respiratory system, keep in mind that your body is always making acids, and those acids must be removed through your urinary system. And if one's kidneys shut down, then you become very aware because the body becomes extremely acidic. You go into acidosis, and that acidity can have dramatic effects on protein structure and cellular function. So we've talked about that pH of 7.4 for blood. We'll get to that again in respiratory and urinary, but you appreciate that the urinary system must get rid of those acids. So here's just a picture of showing this. Yes, nutrients are coming through your digestive system, but right, you've got to be moving those products through the pumping of the blood, sending it off to the tissues. The kidneys and the bladder are receiving those waste products, and of course there's gas exchange going on with the alveoli. That's what that's sort of showing in a cartoon representation. We'll be talking a lot about, at this point, the rest of the course is going to come together, I hope, in a beautiful way, as you're going to see how critically connected the respiratory system is with the urinary system and the cardiovascular system. So these three units are going to come into great focus. Most patients in an ICU, right? Most patients who are very, very ill have issues with their kidneys, lungs, heart, right? Those are the three big body systems that cause the vast majority of huge body problems. Yes, of course, there can be stroke and there can be other things, but those are the big ones when it comes to physiology. So we'll be looking at how these systems all marry together. So I'm going to just jump over. You can read through the review on the structures of the digestive system. That will not be a problem at all. Recall last semester, too, we looked at the layers of the digestive tract. Remember that as food passes through the gut, you have the mucosa. That's the mucous membrane. Then there's the submucosa. Just in that submucosa is where there are lots of blood vessels that are receiving the food. And you will learn in the lymphatic structure conversation that many of your fats are absorbed not into your bloodstream directly, but are instead absorbed into your lacteals, these structures of your lymphatic system. So in that submucosa, there's also a lot of lymphatic structures that's going to help catch organisms that might come into your body through the digestive system. And there's lots of exocrine glands there as well, because you are releasing substances to help break down and keep your digestive system moist from that layer. Then there is the Muscularis Layer. This is a double layer of smooth muscle. One is longitudinal, one is circular. This layer of muscle, these two layers, are helping to keep that peristalsis going. And then finally, the outer layer is the serosa. So keep that in mind. Now, why in the world would we say that there's no serosa in the pharynx, no serosa on the esophagus, or no serosa on the rectum? What does the term serosa suggest? That it has a serous membrane around it. So if the organ is not in a serous membrane compartment, then we're not going to call the outer layer a serosa. Instead, we call it an adventitia. It's really kind of semantics, but the esophagus, can you picture it? It's coming down through the thoracic cavity, down through the mediastinum. the outside of the esophagus would not have on it, quote, a serosa. Remember, the serous membrane going through the thoracic cavity would either be the parietal pleura, which is lining the thoracic cavity, or the parietal pericardium, which is in the wall of the pericardial sac. So there would not be a serous membrane surrounding and lining the esophagus, so we call the outer layer instead a adventitia. It's largely the same thing, it's more semantics, okay? But no serosa instead. That would also be true of the bladder and of the kidneys, because what do we know about the kidneys and the bladder? All of that is retroperitoneal, again, meaning that it's behind the peritoneum, so there would not be a serosa. There would instead be an adventitia. Okay, that's just a distinction in those. Mesentery, this is also a new term. I didn't use this word. What does mes mean to you? Mesoderm, mesothelium, mesentery, middle, right? So here what you have is a double sheet of a peritoneal membrane. We call this the mesothelial layers. You've heard of endothelial. Endothelium is what? The lining of simple squamous inside your blood vessels. You've heard of epithelium, right? And now we have mesothelium. And mesothelium is the term used to describe your serous membranes. So the pleura, the pericardium, and the peritoneum are mesothelial, right? They are simple squamous layers that are in the middle. So if you look at this, when you look at, for example, if you remember what your fetal pigs look like, if you ever tried to separate away the intestines one from another, you would have seen this very thin layer. That's the mesentery, and in there are a lot of blood vessels. And you can kind of see that here. There are mesenteric arteries and veins that are traveling through that very thin layer. It's mesothelium. It's a double layer of serous membrane is what the mesentery is. Okay, moving on. Review this information. There's also some nerve plexuses. It is true that you have a huge number of neurons in your brain, but the second most populated place for neurons is in your digestive system. There's an entire, quote, enteric nervous system, a huge connection of your nervous system with your digestive system. This is all autonomic, nothing over which you're aware, but there is this tremendous nervous activity in your gut. Those nerves are part of plexuses. Right, we know a plexus is a group of nerves that travel together, and these two plexuses are parasympathetic and sympathetic, so they're going to be controlled by autonomic signals, and the two are the submucosal plexus. So where is it located? Right below the submucosa, and this is going to bring nerve endings into the mucosa and the submucosa, and then there's a myenteric plexus, muscle. Right, so the myenteric plexus is a group of neurons that go to the intestine, but specifically speak to the muscle layers of the gut. And they're going to control that peristalsis, that dance that occurs. They're going to be responsible for the timing of the peristalsis and the movement of foods through your digestive system. Okay, so together, these two plexuses are going to work together to keep food moving properly through your gut. Okay, so that brings us now to some new stuff. I've already been here, haven't I? 22.3 was that short section that I jumped to last week because I wanted you to have a better complete story of some differences between smooth, skeletal, and cardiac muscle. So we did look at 22.3 last week. Is there anything about smooth muscle that I can clarify? Anything about smooth muscle? What do we know about it? Involuntary? Shape? Fusiform, typically in shape, single nucleus. But what was the difference here? Smooth muscle could be innervated not by a single axon terminus, but could be innervated in multiple places. And that is one distinct difference about smooth muscle. What about what else was in here about this? No T-tubules. What did that tell you? T-tubules were important for what? They were the rabbit holes, right? The action potential would spread along the sarcolemma, around the muscle, and then would dive down into those T-tubules, and there it would come in contact with the sarcoplasmic reticulum and the release of calcium. There are no T-tubules here, suggesting what? that the signal doesn't travel along the muscle the same way. In fact, there are many places that a neuron can touch a smooth muscle cell, so there is no T-tubule, there's no rabbit trail or rabbit hole for this to occur. There are no myofibrils and there are no sarcomeres, meaning when you look under the microscope, you do not see striations, right? There's no regular arrangement of actins and myosins in the sarcomere arrangement. So that gives the muscle that smooth look. Instead, as we said before, smooth muscle tends to kind of twist up or look at corkscrew, like a little wrinkling. And here's that idea that multi-unit smooth muscle cells. So they're not innervated the same way as skeletal muscle or cardiac muscle. And as a result, there are a lot of gap junctions. Now, gap junctions are similar to intercalated discs. We know the purpose of intercalated discs in the cardiac muscle, similar here in the smooth muscle. And here there are pace setter cells. They're similar to pacemaker cells. So what does that tell us? The smooth muscle will what? Contract without direct connection to the central nervous system. This enteric nervous system, this single unit of nerves, is sort of directing its own peristalsis, right, without the need of the central nervous system to send instructions. We know the sympathetic and the parasympathetic systems can help modulate, right, turn up and turn down this contraction of the smooth muscle, but it does not initiate it. It's the same idea back to the cardiac muscle. The central nervous system does not initiate contraction of your gut muscle. It simply modulates it, increases it or decreases it. Okay, and smooth muscle is tremendously plastic or has a great plasticity, which means when you go to a buffet, right, and you overstretch the muscle, how is that different? What does smooth muscle allows you to stretch without recoiling back? What do we know about cardiac muscle? If you stretch the heart, aka a lot of preload, lots of blood in the heart, Frank and Starling said that the heart will then contract more forcefully. In the smooth muscle, you can stretch the smooth muscle and it does not necessarily contract back in great force. So there's a lot of plasticity, a lot of opportunity for change in the diameter of smooth muscle structures. Same is true with the bladder, right? Every time the bladder fills, it doesn't forcefully begin to contract to fight that stretch. Okay, so same idea. So that brings us back to the idea of tone. Okay, so that brings us quickly through the first couple units and a review on smooth muscle. Again, everything in the digestive system is smooth muscle except orbicularis oris in the tongue and the anus. Everything else is going to be smooth muscle and depend upon smooth muscle for control. So as food is being pushed through, and I really should call it a bolus, right? As you push that mass of food through the stomach, it's going to create chyme. That chyme is that liquidy, pasty substance that's formed by the stomach and the acids of the stomach. And then from that point, it's really just liquidy chyme that's going through the intestines. Those layers of circular and longitudinal muscles are going to be squeezing and doing this dance, as I call it, called peristalsis. And here you can see what happens to food as it goes through. Okay, so here's your bolus of food, and it's traveling toward the end, toward the anus, toward the end of the digestive system. And literally, the muscle layers, first the circular muscles will contract behind, and then the longitudinal muscles will contract ahead of, and that will cause this dancing, right? This wave-like dancing of the muscles, so that chyme and food-like substances, that bolus, will continue to push through. This is largely timed. There's a synchronization to this. It is not a random process. So your gut is always, because of that pacemaker idea, those pace setter cells, there's a certain number of contractions that the gut will undergo every minute and every hour pretty much automatically. Okay? And then there's also, in addition to peristalsis, there is segmentation. And as the name suggests, a small segment of the intestine will sort of churn and fragment that bolus into smaller and smaller pieces, because the ultimate job here is to absorb those nutrients. So we've got to break this stuff down into small, small pieces. Then there are going to be enzymes along the intestine that are going to continue to break down and digest those products and eventually absorb those nutrients. So here you can see this idea of segmentation. Literally, little sections of it are going to be, and those little segments will then allow for this churning and mixing. Okay. So just know there's peristalsis and segmentation. Now, what is going to control this? As you can imagine, there are hormones involved, there's nervous system involved, and this idea of local factors. So as you there are local changes that can occur in the gut. One is pH. We'll talk about the effects of pH. We'll talk about the distortion, the stretching of the digestive muscle wall, as well as certain chemicals that are present that can influence digestion. Then there is also Neuro Control. This makes sense. I've already told you there's plexuses coming down and are involved in the muscle layers. It was the mesenteric reflex. So these are triggered by chemoreceptors, and this has a lot to do with the content of the food, and that's going to start controlling those neurons that are going to push the food through and cause peristalsis to increase. There are also long reflexes. These are going to involve interneurons. So now we're heading back to the central nervous system. Like I told you, the initiation is not by the CNS, but it can certainly modify the movement of your digestive system. This is mostly parasympathetic, right? When the bear is chasing you, then there's less blood flow and therefore less digestive activity going on. And during parasympathetic, more activity is going to be occurring in the digestive system. And then there's a whole host of hormones, a couple of which we'll learn about, that are going to help regulate the movement of proteins and products through your digestive system. These hormones are produced by enteroendocrine cells. So look at those words. Entero means what? Entero means gut, right? Enteric, entero, all those terms mean gut. Endocrine. So these are cells, not so much a gland, not like the pancreas, but little cells, individual cells that themselves make hormones that are going to help regulate this process through the digestive system. So just to get this whole picture in perspective, here we can see that going through the gut, there would be local factors that would be released by secretory cells of the gut themselves or by these enteroendocrine cells. And that would have an immediate effect right back on the local environment of the gut. That would not involve hormones coming from the pancreas. That would not involve signals coming from the CNS. But in addition to those local factors, there's also hormonal controls. And this is largely coming from the gut and from the pancreas, right? Insulin, glucagon, some of those hormones are also involved with regulating the process of digestion. And then finally, the CNS can get involved and send signals down to that mesenteric plexus, which is going to help with moving and setting the pace or modulating the pace of peristalsis and segmentation. So that was just a quick review, and now we're going to head into a section. This section 5 is also one of those review sections. This is just reminding you, we're kind of going from mouth to anus as we go through this, and section 5 is all on the big ideas of the oral cavity, the pharynx, the esophagus, and through the structure. So this should be very straightforward review. On this image, on the left-hand side are the major digestive organs. Again, those are the organs through which food passes. On the right-hand side are the accessory organs that contribute to the digestive process. And you know that ingestion is the taking in of food and water. Mechanical processing would be the churning of the gut, churning of the stomach. You've also got digestion using enzymes, chemicals, and acid as you go through the digestive system. Along the way, we're releasing, secreting products and absorbing useful products. There's also, after you have gone through the absorption process, now the large intestine is largely in the job of compaction. That is to take all those pieces that were not digestible, not usable, and eliminate them through the feces. So all of those are the major issues of the digestive system. Looks like I just skipped over 6 and 7. And I think somewhere I said to skip a couple. Let me go back to the first page. I told you to skip 6 and 7 in the front slide, right? Yep. Okay, so that's good. That's why I just skipped over them. Okay, thank you. I think it was teeth. All right, we're not going to focus on teeth. Okay, so if you're going into dental hygiene, I apologize. So 22.8, okay, jumping over to the pharynx. Again, we're just kind of continuing from the oral cavity. We forgot about the teeth. They chew, and that's about all we're going to care about. And now we're going to head down the pharynx. Remember the pharynx? We learned about 3 parts of the pharynx, the naso, the oro, and the laryngopharynx. Those are the 3 anatomical sections of the pharynx, and they're shown here as a reminder. Again, I'm going to hope that this is all looking really familiar, dusted off. The test will have some questions on anatomy, but largely it'll be on the physiology, which is what I'll be focusing on. So this was one of those sections. I just want to touch those things. Now, the esophagus does have parasympathetic and sympathetic fibers going to it. We see that there are nerves going to the esophagus called the esophageal plexus. That's a generic term, right? Plexus, a group of nerves that go somewhere. The group of nerves that head to the heart, cardiac plexus. The group of nerves that go to the lungs are going to be the pulmonary plexus, okay? So we're going to see this esophageal plexus, simply the nerves that head to the esophagus. You appreciate that once you swallow, you do not have voluntary control over the rest of the process. So the rest of the esophagus is mostly smooth muscle. There's a little bit of, as you initiate swallowing, the very top of the esophagus does have some skeletal muscle as you begin that process of swallowing. But shortly thereafter, it's almost all smooth muscle. So you don't have any control over that. That's why you need this esophageal plexus, which is helping to do that peristalsis and push that food down. Of course, you've got gravity to help you, but you can swallow, right, when you're upside down. And you can certainly, the muscles will continue to push food toward your stomach, even if you are upside down. There are 2 sphincters here. In 105, we learned about the cardiac sphincter, also called the lower esophageal sphincter. And no one ever asked me last semester, but maybe you were thinking, well, if there's a lower, there must be an upper esophageal sphincter. And there is. This is at the very, very top. It's a band of smooth muscle that functions like a sphincter. Remember, sphincter is a circular muscle group. And it's going to prevent air from entering the esophagus. Now, babies have issues with this, right? When they're suckling, they get air down their esophagus. We have to burp them. We get control over this sphincter. The lower esophageal sphincter is the one dropping into the stomach at the region of the stomach called the cardia. What are the layers here? Again, mucosa. And what kind of epithelium do we find here? I'd want you to definitely go back and remind yourself of the histology as we go through the different regions of the digestive system. So the oral cavity and the esophagus are lined by non-keratinized stratified squamous. Recall that's multiple layers, flat on the surface, but living, right? Moist and living at the surface. And then there's going to be a layer of muscle. And then, like it says here, the top third of the esophagus is skeletal muscle. The middle third is a mix, and the bottom third, roughly, is 100% smooth muscle. And there is no serosa, as we've already described. There would instead be a adventitia. It's semantics, but it's an important one. All right, it's an important semantic. So as you look at the esophagus, it could be, what else could it be? It could be easily confused with the small intestine, to be honest. Okay, you still see that ruffled appearance of the mucosa. You see a thick muscle layer, the muscularis, a couple layers of muscle. You still see an outer layer, the adventitia here. So it would be easily confused with the intestine until you look carefully here at the mucosal layer. And you see here not what would be in the intestine. In the small intestine, the mucosal layer would be simple columnar. Okay? And you see lots of microvilli there if you could see them under the light microscope. Here, when I zoom in and I see the mucosal layer, I am seeing what? There's my stratified squamous, and at the surface I see nuclei. So that would be the non-keratinized type. When you're swallowing, there are 3 phases of swallowing. We all do this every single day. Think about it. You choose, when you have food in your mouth, you choose to swallow most of the time, right? You chew the food up, and you've got that piece of food in your mouth, and you choose to break it up into 2 or 3 pieces and swallow it. That's initiated as a voluntary action. But as soon as you begin to swallow, it quickly becomes an involuntary reflex. So the very first part is called the Buccal Phase, or the Buccal Phase, usually buccal. And this is a 100% voluntary phase. Now, that is not to say that right now some of you are swallowing involuntarily as you are swallowing your secretions, right? But here I'm talking about as you're truly thinking about swallowing and as you're eating, you're definitely thinking about it. There's a voluntary feature to this. At this point, you're making that bolus. The tongue is helping to push the food up. You're pushing the food back to the oropharynx. You're going to block off the nasopharynx by the lifting of the uvula, and now you're going to start pushing that food down into the oropharynx. The pharyngeal phase is basically when you hit the uvula. All right, when you hit the uvula, you're back there at sort of the border of the oropharynx and laryngopharynx. At this point, the medulla oblongata kicks in. And the medulla oblongata, as you know, is part of your brain stem and is involved with regulating autonomic things. So the medulla oblongata will pick up the responsibility and is what is going to fold the epiglottis, right? And help the soft palate to elevate. You don't think about that. That's happening for you as part of this reflex. Individuals with brain damage, individuals with damage to their brain stem are going to have issues with swallowing, and they'll lose coordination of this, and they'll be choking all the time. And as a result of their choking, they'll aspirate food down into their lungs. So we recognize that brain damage, especially the medulla in this brainstem area, can have devastating effects on the ability to swallow in a coordinated way. The last phase is the esophageal phase, and at this point, we're way down. We're in the esophagus. Bolus is being pushed down by peristalsis. Again, the top is somewhat skeletal muscle, but the bottom of it is completely smooth muscle. About 9 seconds, right? About 9 seconds average to get food from the top to the bottom of the esophagus. If you've ever had something stuck, you know you can actually kind of feel it moving down. Liquid is certainly going to move a lot faster. And if you do have a dry throat or a dry esophagus, it could certainly take a little bit longer and require a little bit longer, right? You'll actually feel it kind of get stuck, and you'll feel things kind of work to get it down. So let's just go through those 3 phases. Okay, the buccal phase, voluntary. The pharyngeal phase, the beginning of the involuntary control by the medulla oblongata, and then the esophageal phase, when truly it's just peristalsis pushing things down. Okay, so now let's talk about the mesenteries. Okay, now this is relatively new. We're now down in the peritoneal cavity. The peritoneal cavity, remember, is the serous membrane that's going to envelop all of the intestines, or most of the intestines, and your abdominal pelvic cavity. And there's 2 parts to this. This is review. Excuse me, there's the visceral peritoneum and the parietal peritoneum. Remember, the visceral layers are shrink-wrapped to the surface of all the organs, and the parietal layer would be lining the inside of the peritoneal cavity. What is a serous membrane? It is a simple squamous layer. You might see it called mesothelium. And the other thing here is that there's fluid always here in this cavity. There's about 7 liters a day that you're secreting and reabsorbing. That's total, right? Production and reabsorption. At any one moment, you might have about 50 mils of this stuff, 50 milliliters of this fluid, and that's in between those layers. So if you think back to pictures of the peritoneum, you'd have the double layer, and in between the layers there's fluid, right? Peritoneal fluid. This is that 50 mils. And of course, that's going to help reduce friction, allow your internal organs to slide over each other. You can do jumping jacks, all that kind of stuff, without worrying about your organs getting caught up on each other. The rate of fluid movement can be accelerated by liver disease. We've talked about this before. What does the liver make? If you have a malfunctioning liver, the liver is making the albumins. The albumins are the primary regulator of fluid levels. And if you have too much fluid accumulating in your gut, we would call that ascites. If also your kidneys are diseased, what's going to happen? You're not going to be getting rid of fluid as properly as you should, and that fluid will back up and again lead to abdominal fluid buildup, and or there could be problems with your heart. And if you've got issues with your heart, especially one side over the other, you could have fluid buildup in areas because the blood's not being sucked back to the heart appropriately. Okay. So all those systems can lead to fluid accelerating building up in the peritoneal cavity. So the mesenteries, these are those layers. They are formed during your embryological development. They're basically separating and connecting and holding in place all of your digestive organs. When you look at the gut, you're always impressed perhaps by, gee, how is it that the ascending and transverse and descending colon are so properly arranged? Well, they are being held in place by these mesenteric membranes. So there is, on the what will break down into dorsal and ventral. The dorsal mesentery developmentally becomes the greater omentum. So the greater omentum is a term you need to know. Omentum means fat, and it's this basically an apron. The greater omentum is really this big apron that connects the stomach to the intestines. It forms this big pouch, and mostly is fat. Remember that the gut organs are not protected by any sort of, really any kind of skeletal system. Your gut needs some extra layers of muscle. There's multiple layers of abdominal muscles, and there's this greater omentum that creates a packing layer of protection. There is itself the mesentery proper that's going to connect the small intestine to the body wall, and the mesocolon, which connects the large intestine to the body wall. So the mesentery proper is small. The mesocolon connects the large intestine. There's also the ventral mesentery, which becomes your lesser omentum. This is going to connect the stomach to the liver. So that's what I want you to know. The greater omentum is stomach to small intestine, right? And the lesser omentum is the stomach to the liver. These mesenteric layers also provide a layer through which blood vessels and nerves can travel and be protected. There's also the falciform ligament, and this connects the liver to the anterior body wall. And you'll see that falciform ligament when we look at the liver. The liver has 4 main lobes, and you'll see this ligament sort of separate, in part, the liver into its lobes. So here we see sort of a cross-section, and let's take a look at the major ones. The greater omentum was what? Connecting the stomach to the intestines, right? Small intestine. And then the lesser omentum was connecting the stomach to the liver, and the falciform ligament was connecting the liver to the wall of the peritoneum. So you see those 3 structures, connective tissue structures, holding these digestive organs in their proper location. From a different perspective, again, here's that falciform ligament coming into the liver. Here's the lesser omentum connecting the stomach to the liver. The greater omentum connecting the stomach to the intestines. You can see the pancreas is shown here in this cartoon. The mesentery proper is helping with the small intestines, and the mesocolon is what's connecting the large intestine to the wall. So these are just five or so new structures that are anatomically holding everything into place. Collectively, they're referred to as the mesenteries. These mesenteries are going to, again, help position everything. They're going to contact and fuse different structures together, and much of the mesocolon fuses with the dorsal body wall. So it's not something you can see so much, but it's what, when you look at the large intestine, you'll see that the large intestine is largely fused to the backside of the peritoneal cavity, and that is the mesocolon that is connecting it all together. This might be an easier view to appreciate. This is one you've seen probably more familiar. Here, the greater omentum has been cut. It would have been sort of a big pouch that would come up and over the stomach and come down to the intestines, the small intestine. And then the mesocolon is going to fuse and hold that large intestine to the wall. And the lesser omentum, again, was the connector between the what? What's missing here? This right here would be the liver, right? So that's the connector between the liver and the stomach. And then you can see the transverse, the ascending colon are all held in place by these mesenteric membranes. one more view. Okay, so what do I have? Attaching the liver to the wall, falciform ligament. Connecting the liver to the stomach. What was that? Lesser omentum. Connecting the stomach to the intestines. Greater omentum. And the mesocolon was connecting the what? Large intestine back to the wall. And the mesentery proper was connecting the small intestines to the wall. See the mesocolon up here as well as down here. Okay, so those are the mesenteries. It's just looking at 4 or 5 structures. The stomach, also this is a review section. I'll go through and look at the highlights of this. Recall that the stomach is going to be receiving the food first into the area called the cardiac region. And there are 2 curvatures on the stomach. You know the stomach is sort of a J-shaped organ. The lesser curvature forms a medial surface. This is going to attach to the lesser omentum. The greater curvature is going to be more lateral and inferior, and this is going to attach to the greater omentum. So that's easy. Lesser curvature to lesser omentum, the greater curvature to the greater omentum. 4 regions of the stomach. Fundus, that's the superior portion, that's the dome of the stomach, recall. The cardia is the first few centimeters of the stomach, and here this is going to start producing, this region of the stomach is important for producing mucus. Now, why would we need mucus produced here? It tells us that cardia, if you have acid reflux or something that's gurgling up from the stomach into the esophagus, the stomach is designed to withstand that low pH and that acidity, but the esophagus is not. So there's always going to be some reflux, a little bit going up, and you want a lot of mucus to be traveling back up into the esophagus to protect it, to line it from that acidity. So the cardia, those cells that make up that region, are making a lot of mucus to help provide protection to the esophagus. The largest part of the stomach is the body. That's the majority of it. And then finally, you've got the pylorus, the very bottom region of the stomach that is going to be taking food into the small intestine. The word pylorus means gatekeeper. So it is the last part of the intestine, of the stomach, that's going to be, quote, gatekeeping the movement of this chyme into the intestines. So this is a nice view. I like this. We're coming down. Esophagus is coming down here. You can kind of see it dropping in. Now, from lab today, those who had lab today, this esophagus drops through the diaphragm because of an opening called the esophageal hiatus. Okay, you'll see that in lab today. The hiatus is a break. It's a break in the diaphragm that allows the esophagus to come down and break through the esophagus, or through the diaphragm. That's not shown here. And then here is that lesser omentum. Again, what was it doing? Stomach to liver. You can see it very clearly here. And then this is the greater omentum, and it's this big, fatty, like shield, almost like a big apron, that's going to connect the stomach down and over the intestines. Don't forget, we've also got up here the spleen, right? It's tucked up here next to the stomach. That'll become important in your lymphatic conversation. We've got the little gallbladder sitting over here. Anatomically, again, this is all real, but you can see here this, again, the curvature is here, right? The lesser, this is the lesser curvature. And what is it up against? The lesser omentum, which connects to the liver. And then the greater curvature is what's up against the greater omentum connecting down to the intestines. Layers here are similar, except in the stomach there's not 2 layers of smooth muscle, there are instead 3. In addition to the circular and the longitudinal layers, there is an oblique layer. Now these 3 layers of muscles can really churn and mix the food. Along the stomach, there are rugae. Again, this is a review, wrinkles that are helping to increase the surface area of the stomach. And then when you're down to the pylorus, there's that big cavity area, sometimes referred to as the antrum. There's the canal that's going to empty directly into the duodenum, and there's the sphincter in between. That is the pyloric sphincter. So the big area down here is the, the antrum, if you will, of the pylorus. The canal would be the very last little bit going into the sphincter. 3 layers of muscle, right? Longitudinal, circular, and oblique. And why not? Let's do it. This is what? The greater? Let me go back. This is the greater curvature because it's connected to what? Greater omentum going to the small intestine. This is the lesser curvature because it's connected to the lesser omentum connecting to the liver. Have I killed that enough times? Okay. Killed, done, got it. Must be a couple questions on the test. Not that hard. Bonus points, right? Not bonus, just easy, straightforward. Remember my voice. Okay. Let's get into the functions, really the physiology. So we know the stomach breaks down food. It's going to mechanically break it down. There's going to be acids and enzymes added to the food. And there's also in the stomach the production of intrinsic factor. Intrinsic factor is an incredibly important molecule that is necessary for the making of vitamin B12. So let me get to that in a few minutes. But intrinsic factor is very, very important. So let's go back. The layers, again, mucosa, submucosa, muscularis, serosa, right? Got it? Been there, done that. We know the layers histologically. The span of these cells, they only live for about 3 to 7 days. That makes sense, right? These cells are constantly replaced because they are constantly being churned off as well as exposed to acid. So that it's pretty hostile, right? Pretty hostile environment. And so those cells are constantly being replaced. Here's a quick cartoon of this, and all this bracketed area would be the mucosa, going down into those rugae, into those grooves, and down into these gastric pits. And down in those pits are where the epithelial cells are that will make the enzymes and make many of these substances that are involved with the digestive process. Now, each of these, let me make sure we're okay with this. This finger-like structure that I'm going around is a villus, right? A villus. It is this big finger-like extension. And then what is on the surface? What would we expect to see? Not so much here, but in the small intestine, we would see microvilli. So the villus is the big fold. The microvilli are the small extensions that are involved with surface area. And then down here, also to bring back that idea, the mesenteric plexus. So down in that muscle layer is the mesenteric plexus. That is the group of nerves that are controlling the muscle layer of the gut. There are lots of gastric glands. These glands are producing the enzymes and the acid that are important in the stomach. The cells that make this are the parietal cells. The parietal cells and the chief cells. Together, they're making about 1.5 liters of gastric juice every day. There's also glands here in the pylorus that are helping to regulate the movement of food through and help to coordinate with the hormones the movement of food. We can appreciate that digestion needs to be highly orchestrated. You want food to pass through slow enough for absorption of nutrients to occur and for digestion to be complete. And you want food to pass through fast enough that it doesn't rot and cause bacteria to start building up gassy things. So we want this to all be beautifully coordinated, and that is in part by these gastric glands and their secretions. The pits are the deep areas, and there are stem cells here. Those stem cells, again, are always coming to the saving grace of your stomach because it is constantly shedding off the more superficial cells. So these little gastric pits and gastric glands are talking to each other. Now, what's being made here? In the gastric glands, there are parietal cells. Boy, that word parietal is everywhere, right? We've got parietal lobe of the brain. We're going to see parietal in a few other places. I usually say that parietal means wall, right? And it does. But these are also called parietal cells. They are the ones secreting intrinsic factor. And intrinsic factor is hugely important in helping you absorb B12. If you didn't have a stomach, or you had gastric bypass, and a large part of the stomach has been removed, then that person will not make adequate intrinsic factor. And if they're not making adequate intrinsic factor, then they will not be getting absorption of B12. Pills or injections, okay? And then B12 goes back to the blood unit. And B12 was critical for the making of red blood cells. So a person following gastric bypass typically will be B12 deficient, and that can lead them to anemia. So they need to be watching their B12 levels, taking injections or supplements. But again, if they take in the supplements, their stomach is already sort of, been compromised, and so oftentimes injections are necessary. Also, the parietal cells, in addition to making intrinsic factor, are making the hydrochloric acid. So these are the cells that are making the acid. This acid that's made here is going to activate pepsinogen. Now, where have you seen proteins that end in ogen? Remember fibrinogen? Fibrinogen was the protein that's always floating in your blood. And what happened? Fibrinogen would be cleaved or used to form fibrin. So the proteins that end in ogen are pre-made but not yet active. So when HCl is made, it activates pepsinogen. Pepsinogen is then cleaved to make pepsin. Pepsin is the enzyme that's cutting many of your proteins down. But it's sitting there inactive, right? So the HCl is necessary. So if you don't have a lot of acid, then you would not be activating the pepsinogen to make pepsin. Your stomach pH is somewhere under 2. Okay, that HCl is keeping that pH really quite low. There are also, in these gastric glands, G cells. And these are those enteroendocrine cells that I mentioned. And they're called G cells because they're in the gastric pits. And they're secreting a bunch of enzymes as well. Okay, so we've got parietal cells, G cells, and then finally there are chief cells. The chief cells are making the pepsinogen themselves, right? So the pepsinogen is made by the chief cells. The parietal cells made the acid. Together, they're going to activate that pepsinogen to be cleaved into pepsin. And then pepsin is a huge proteolytic enzyme, which means what? An enzyme that lytic breaks down proteins. So when you absorb proteins in your body, those proteins are broken down into their individual amino acids before they are absorbed into your gut.
So the HCl breaks down the
Pepsin, and then the pepsin is the enzyme necessary to break down your proteins during digestion. Now, the problem with the word pepsin is that it doesn't end in ace, right? You've sort of been trained by me and others, perhaps, that proteins that end in ace are enzymes. And that's true. Pepsin is an enzyme, but it is not ending in ace. It does end in IN. It is a protein. Remember, proteins and enzymes are connected, but it is an enzyme. Now, these cells are also producing gastric lipase. Gastric lipase is important for babies to break down milk. Okay? So, not as important. Adults sometimes have problems with breaking down milk, in part because they don't have that gastric lipase. Okay, so those are the cells. Where are we? Let's make sure we know where we are. We're in one of those pits of the mucosal layer of the gut, of the stomach. And down in those pits, there are cells. Some of them are parietal cells, some of them are G cells, and some of them are chief cells. I'm not going to ask you to know the difference from looking at them, but those different cell types are down in those deep gastric pits, and it's there those important substances are made. And those substances then come up to the surface of the stomach, right? They come up this tube. So we would call these exocrine or endocrine. Exocrine, right? Because they're being released to an epithelial surface. And while we don't think about it, the lining of your gut from mouth to anus is all outside of your body, right? From mouth to anus, you're really just a tube going down the center. It all has access to the outside world. Therefore, anatomically, your gut tube is actually outside of your body from an anatomic standpoint. Now, HCl. The parietal cells do not create HCl in their cytoplasm Otherwise, that acidity would destroy the cells themselves. So that acid is released, secreted out into the tube. So what has to happen is that hydrogen, H+, and Cl- must be transported separately. Otherwise, they would combine to make HCl. Right? So there's got to be two different mechanisms. We've got to get acid in there, or we got to get H+ in there, and we have to get chloride ions in there. Here's an enzyme that you're going to hear about over and over and over the next few weeks, and that is carbonic anhydrase. Okay, carbonic anhydrase. And this is an enzyme, ACE, that takes CO2 and water to make carbonic acid. So this reaction right here, the enzyme necessary for this to occur, that arrow, is helped out by the enzyme carbonic anhydrase. H2CO3 is carbonic acid. Okay, so CO2 plus water make carbonic acid, H2CO3, with the help of this enzyme called Ca, carbonic anhydrase. What does that carbonic acid do? It then breaks down an HCO3, that's bicarb. Okay, so bicarbonate is the HCO3, and H plus is the, remember H plus, when we think about H plus, we think more H plus, the more acidic something is. So this H plus is what's going to be combined with Cl minus in a moment, right, to make HCl. But we've got to get this H plus from a different place than we get the Cl minus. Otherwise, they would combine and make acid inside the parietal cells. Now, the bicarb is going to be ejected from the cells and will be exchanged for the chloride. So that, what's the charge on bicarb? Let's go back. What's the charge on bicarb? Negative, right? -1. And chlorine is also -1. So these guys kind of exchange, if you will, and this is going to bring the chloride ion in. And from here now, you're going to be able to create this HCl. Now, if the from the interstitial fluid, the bicarbonate can enter the bloodstream, and there's a lot of bicarb. Bicarb actually makes things more alkaline. Okay, so bicarb, what do you take when you have an upset stomach? It's Tums, and if you look at the active ingredients, there'll be some bicarbonate in there. So the bicarbonate is an alkaline substance. When you are treating a hot tub or a pool, you'll throw in some bicarb, right? Basically baking soda is what this is. It's really, think about it that way. It's sort of like baking soda. And that's going to help neutralize the acids of your stomach as well as neutralize the acids of a pool. So the bicarb can actually drive the pH of your blood up. Does that make sense? Up because it's alkaline, and things that are higher in pH number are less acidic, more alkaline. So as a result of this, we won't get into all the physiology of this, but basically what you see is that hydrogen is released into the lumen. Here's your cell, and this would be the interstitial fluid. And do you agree that wherever there's interstitial fluid, that's the fluid between your cells, really close by there's going to be a blood vessel. So I've just drawn the blood vessel in, right? That wasn't shown here. So you're going to have bicarbonate that's going to be entering in the bloodstream. And what you see is that this is now the lumen of your gastric gland, and your stomach lining is here. And so you've got chloride being brought in, you've got hydrogen being brought in. They're being brought in by two different mechanisms. Those hydrogen and chloride ions are then meeting in the lumen of the gastric gland, and that's forming the HCl. Again, if the hydrogen and the chloride were combining inside the cell, it would be damaging to the cell itself. So just know there's three different cells, right? Parietal cells, chief cells, and G cells, and what they're producing in the gastric pits. What else do we have here? As we go through the intestine, there are a few other special things. And again, we know this. There are folds. The intestine is incredibly high in surface area. There's a lot of folds that are inside your small intestine. This is going to increase the surface area. 800 folds in your small intestine, mostly in the jejunum. So the jejunum is what part of your intestine? First, middle, or last? last part, right? No, sorry, middle part. Woo, don't misspeak, Sean. The first part was the duodenum. The middle part is the jejunum. The last part is the ileum as you go toward the large intestine. So the jejunum is the middle part of the small intestine, and that's where most of these folds, right, are found. Now, I mentioned this already. Those individual folds of the intestinal wall, each of them is called a villus, right? Each one's a villus, many villi. They are themselves finger-like projections of the mucosal membrane. But then covering the villus is an epithelial layer, which oftentimes has microvilli. All right, so don't get those two things confused. The microvilli, recall, are things you cannot see under a light microscope. Very, very small little, sort of like a pleating of the surface of the cells to increase the surface area. So you've got the folds of the intestine plus the microvilli. So we're really increasing the surface area drastically by a factor of over 600. If the intestine were a smooth tube versus the folds and the microvilli that are there, you've got a 600 increase in absorption area, which allows digestion to go faster, right? It allows things to go through faster to increase those substances as we're moving through. There are glands in the intestine called the Crypts of Lubricant, okay? And these crypts are basically stem cells that are also making glandular secretion. So not only is the stomach releasing different substances, so too are the intestinal cells from these crypts, right? Again, it's a guy's name, so we're stuck with it. Some German guy, right? Leberkuhn or Swiss guy. And these cells are going to be shedding and constantly replacing themselves and secreting substances. So when I look at this could almost be a picture of the dermis. Do you agree? If I didn't look at all the rest of the labeling. Because what do I see here? Those finger-like extensions. It almost looks like the interface of the dermis with the epidermis. Now, the purpose of that in the skin was to increase surface area so that those two layers don't shear off from each other. Here, you have those big papillary-looking structures. Each one of those is a villus, right? So each of these is a villus, and layered on top of that would be microvilli. The layers are the same. We know our layers. The mucosal layer would be all of those villi structures. Submucosa just below blood vessels and lymphatic vessels. The muscularis, the two layers of muscle, right? One circular, one longitudinal, and then the outer layer called the serosa, since this is in fact surrounded by a serous membrane. Each villus, what is this thing? So we're going to break down this villus. You'll hear discussion of this a little bit in the lymphatic lecture as well. So inside that villus, there's an extensive capillary network, and that is what's called the lamina propria. What does lamina mean to you? Layer, right? So the proper layer, if you will, the propria. And here is where the blood vessels are. These capillaries are picking up the nutrients, and those nutrients are going into your hepatic portal circulation. The hepatic portal vein does what? It's taking nutrients from the gut that have just been newly absorbed and bringing them to the hepatic, to the liver. And the liver, as we'll talk about in this unit, is then going to detoxify those nutrients and make sure there's nothing in there that's going to harm you. Once the liver has done its detoxifying, then those substances are released out the hepatic vein and then travel to the bloodstream. But in addition to blood capillaries that are bringing nutrients to the liver, there are also lymphatic capillaries. These are called lacteals. And those lacteals are actually helping to pick up fatty acids. So there are a number of fats that simply cannot enter directly into your blood capillaries. Instead, those fatty molecules are absorbed into these lacteals. Stacy asked a question earlier about cancer cells metastasizing and traveling through the lymphatic vessels. These fats are first traveling through your lymphatic vessels, but then all of your lymphatic vessels eventually merge back into your bloodstream. So things traveling through your lymphatic vessels are in constant circulation with the things traveling through your vascular system, as well as interchanging with things in your interstitial fluids. Now, along the surface of the villus, there's a little bit of smooth muscle. That's called the Muscularis Mucosa, not to be confused with the Muscularis externa. So you'll see two layers of muscle here. The muscle layer that you have been introduced to mostly was that big, thick, double layer of smooth muscle. That is external. That's the one responsible for peristalsis and significant movement of the gut. This Muscularis mucosa is a muscle layer of the mucosa. It's actually a very thin layer within the mucosa, and this is going to help squeeze just the lacteals and those capillaries and just keep lymphatic fluid moving and keep this process of absorption occurring. There's also along the border of the lacteal and along the border of the villus a carpet of microvilli. Not only does the microvilli increase the surface area, but embedded in those microvilli structures are enzymes. Those enzymes are important for the breaking down of nutrients, and collectively, that is called the brush border. So you see the term brush border here. Brush border is that carpet of microvilli in which there are enzymes embedded. So if we take a look at this again then, this is a lacteal, right? We're looking at one of these humps lining the intestine. So there's the lacteal, and as you look inside there, you see red, blue, and green, and yellow, right? There's some nerves in there. There's green in there, lymphatic capillaries. You see red and blue representing arterial and venous blood flow. On the very surface of this lacteal, there would be cells, and I'm seeing some beautiful goblet cells. You see the nice big clear whitish goblet cells there, as well as the lacteal going up the center. Along the outside, microvilli, which we cannot see on this light microscope, and we would call that outer layer the brush border. It contains not only the microvilli, but a bunch of enzymes important in the digestive process. Any questions so far? Lots of review, some new facts to add to your understanding. Remember the digestive system, at least in my 105 class, is one of the systems we did not directly discuss in lecture. Right? We only saw a discussion of it in lab. You had a take-home exam that covered a little bit of physiology, mostly just the anatomy of the digestive system. So for most of you, this is a new conversation, but the anatomy of this should be familiar. Along the small intestine, this is where most of the absorption of the nutrients will occur. The last 10% will be in the large intestine. The vast majority of your absorption is in the small. This is about 20 feet, right? 6 meters, 20 feet or so, 18, 20 feet. You'll see different numbers in different books. But this is by far the longest portion of your digestive system, with diameters ranging from about 1 inch to about 1.5 inches, just rough numbers. Coming off your stomach, it's about 1.5 inches diameter. So it's a good-sized tube coming off your stomach into your intestines. The 3 sections we've already said: duodenum, jejunum, and ileum. Recall the spelling of ileum so you don't get it confused with the bone portion of the os coxa. And I think we got this. So at the very beginning, the stomach, right? The stomach is right here, basically, and we just dropped into it. The blue portion would be the duodenum, right? See the blue portion? I know your notes aren't in color, but you see the blue portion. Then the bigger portion would be the purple region. That's the jejunum, where most of the folding is. Notice the duodenum is rather short. This is not 1/3, 1/3, 1/3. The duodenum is a relatively very short section. And then after you get past the jejunum, then all the orangey, flesh-colored regions are the ileum. The ileum is then what makes contact with the cecum going into the large intestine. So let's go through this. The first 10 inches, right, of those 18 feet, the first 10 inches or so is the duodenum. This is really where, what's coming into here? Do you recall from the digestive models from last semester? What is dumping into the duodenum? Not only the contents of the stomach, which would include acid, but also right there is the gallbladder is dropping in, as are the pancreatic secretions. All that is dropping in right there at the duodenum. As soon as those acidic substances drop into the duodenum, something has to happen. The esophagus can't handle acid, neither can the duodenum. So part of what the pancreas is releasing is alkaline secretions, because those stomach secretions that came out with the chyme from the pyloric sphincter must be neutralized. So there has to be some base that's coming into the place here. This is mostly retroperitoneal. If you look, this little portion of the duodenum is sort of back behind. Again, the digestive secretions of the liver, the gallbladder, and the pancreas are all dumping into this place. And your biggest job here is not absorption. The biggest job here is simply to mix those pancreatic juices, the gallbladder, and the liver juices, and the stomach juices together. That's really the big job of the duodenum. The major job of the jejunum, with all the folds, was absorption of nutrients. So here's the mixing bowl, right? That's what the duodenum is. Then we get to the jejunum. Again, most of the absorption's happening here. This is the middle 8 feet or so. This is completely contained within the peritoneal cavity, has lots of folds, as we said before, and most of the chemical and nutrient absorption, chemical digestion and nutrient absorption is occurring here in the jejunum. The last bit is the ileum. It's going to end at the ileocecal valve going into the cecum. Fewer folds, right? Most of the digestion is done. Very few toward the end. The villi get shorter. They get more stumpy. So there were long villi in the jejunum and much shorter, if any at all, villi in the ileum. Also, here in the ileum, lots of lymph nodes, right? Submucosa contains aggregated lymph nodes. Again, a big connection between the digestive system and the lymphatic system is that lining your intestines, especially in the ileum, are a group of lymph node-like structures that we'll call the MALT, right? M-A-L-T. So when you hear about MALT, mucosa-associated lymphatic tissue, that is this referring to this lymphatic nodules. So if you were to just have a piece, a chunk of duodenum versus jejunum versus ileum, the thing you would notice, I would say, the number one thing you would notice is the large number of folds, the very ruffled appearance of the jejunum to increase the surface area. Those folds would also include the villi, wouldn't they? And then what we cannot see, of course, are the microvilli. In the ileum, what you would notice, perhaps, is fewer folds and not as deep, right? Not as deep. And actually, as you look over here in the duodenum, I don't see as many folds at all. And I would never ask you to find this, but here in the duodenum is the duodenal ampulla. There was another name for that duodenal ampulla. I think the Ammerman would call it the hepatopancreatic ampulla, right? Last semester, and I gave you the alternative name of the duodenal papilla. Recall papilla as a nipple-shaped structure, so dropping into the duodenum where the pancreas and the gallbladder drop in, that's called the duodenal papilla. So those are the regions of the intestine. Again, hopefully still mostly review with a few new facts mixed in. The next little section is going to go into brand new stuff on the hormone regulation of the digestive system. There are 5 hormones you need to keep track of. It's not too bad, right? 5 hormones here. 4 of them are produced by the duodenum. Okay, so what we're going to see is that the duodenum, because it produces these hormones, is really the regulator of the digestive structure. Once that food, that chyme, drops in from the stomach into the duodenum, these secretions and hormones are going to really regulate the process. The first one is gastrin. Gastrin. Okay? Secreted by the G cells. So the reason they were called the G cells, this was up in the stomach. Remember we saw what? We saw parietal cells, chief cells, and G cells. The G cells were in the gastric, right? They're in the stomach, and they are producing this gastrin hormone. This hormone is also produced by some of the enteroendocrine cells of the duodenum. This gastrin is stimulated by food presence in the stomach. So as you eat, as food arrives in the stomach, it is going to upregulate, kick up the production of gastrin. This gastrin is then going to increase your stomach's mobility, right? Start it moving and churning, and also make sure you're making plenty of acid and enzymes to digest the food. The second hormone is secretin. It is also released by the duodenum as chyme arrives into the duodenum. This is going to upregulate the secretion of bile from the liver and bring in more of the buffering from the pancreas. Remember, the chyme is coming in very acidic. The duodenum can't handle that, and so we have to neutralize that acidity by increasing the pancreatic bicarbonate, right? Remember I told you bicarb? What's being released by the pancreas is largely bicarb, an alkaline substance. But secretin actually decreases gastric motility, okay? So we have gastrin that increased gastric motility, then we have secretin, which is going to sort of slow it down a little bit. Number 3, gastric inhibitory peptide, GIP. This hormone is released by the duodenum when fats and especially glucose enter the small intestine. It inhibits overall gastric activity and directly increases insulin. So if sugar just dropped into my duodenum, what do I need to do, folks? Why do I need insulin? Insulin tells the body, your cells of your body, to do what? Receive and accept that insulin. So as sugar comes into your duodenum, you're going to make more GIP. GIP is then going to help increase your insulin levels. There is amazing research being done right now on GIP. It's amazing how much we're beginning to understand this and how important this is for regulating as a control mechanism in diabetes. Okay, so this is another mode of regulation in some type 2 diabetic injectables. They're looking at GIP. They're affecting this activity. Not only is it going to increase insulin by your pancreatic islets. Remember what cells? What cells make the insulin? Not the alpha cells, but the beta cells, right? The beta cells are the ones being activated to make the insulin. It's also going to increase your lipid synthesis, increase glucose uptake by skeletal muscles as well. Number 4, CCK. CCK is secreted by the duodenum also when chyme arrives, especially if that chyme has a lot of lipids in it. So if you've had a fatty meal, you're going to have more of the CCK. This is going to increase enzyme production by your pancreas and cause contraction of your gallbladder. What does the gallbladder release? Bile. And bile is important for the breakdown and emulsification of fats. So if you have a lot of fat in your diet coming in from the stomach, drops in the duodenum, duodenum makes CCK. CCK basically tells the pancreas, squirt, right? Give me some more bile. I need to break down this fat. Finally, there is VIP, vasoactive intestinal peptide. This is also released by the duodenum, stimulates secretion of your intestinal glands, dilates capillaries, right, to increase the absorption of the nutrients as they're coming into the gut, and inhibits acid production of the stomach. So these are the 5 hormones of major digestive regulation function. And they're all nicely put on one image here, 2214. And here they are. Now, of those 5, which one was not made by the duodenum? Gastrin, right? The other 4 were made by the duodenum. Gastrin was made by the G cells of the gastric pits. Okay. The other 4 were made by the duodenum, and that's shown here nice and clear. Gastrin in the stomach, the other 4, GIP, CCK, secretin, and VIP, all coming from the duodenum. They're each involved with something a little bit different, aren't they? Depending upon the contents of your food, depending upon what's entering into the stomach and into the duodenum, they will increase in different amounts. overall, though, they're all regulating nutrient absorption, right? That's their overall function collectively, is to make sure that you're absorbing the nutrients that you need. And that, of course, is going to be happening in the jejunum, where you're going to be doing most of that absorption. Questions on the hormones? A little memorization there. Not too bad. Okay, so know your 5 hormones. Let's take a little break. Okay, let's take a 5-minute break and revitalize your brain and maybe get some nutrients. Turn your CCK on or something. Any questions so far? Any questions so far? I mean, we're zipping right through this quite well, aren't we? If I look ahead, what do we have? Yeah, we got a little bit left, but we're gonna be zipping through this. We might just finish up the digestive system in the next half hour or so. But anything so far? The 5 hormones, what are they? Gastrin, made by the stomach. The other 4 are made by the duodenum. They are secretin, GIP, VIP, and CCK. Okay, and just what you want to look at is what is it that turns them on, and when they're turned on, what do they do? And usually, what turns them on makes sense. So, like CCK is turned on by fat, and it's going to tell the gallbladder to squeeze and release bile. So they kind of make sense. If sugar comes in, then you're going to increase insulin. If proteins come in, they're going to increase other things. So it does make sense. Now, there are phases to secretions, and the names are according to the location of the control. So there's a cephalic phase. Therefore, it's controlled by the what? By the head, right? And this is when you see, smell, taste, think of food. This is directed by your central nervous system. This is going to prepare your gut to receive food. It's going to stimulate your submucosal plexus. That was the group of nerves around your mucosa layers, stimulated by your vagus nerve. Remember, this is all vagus, right? The vagus nerve is the one that wanders down to your gut and sets up your parasympathetic signals. And this is going to increase your gastric juice production up to 500 mils an hour. I mean, half a liter an hour. That's a fair amount of juice. And this is only going to last for a few minutes. Then you get into the gastric phase. That means that you've actually taken in food, right? You've more than thought about it. You've actually ingested, and the food is now in your stomach. And you're going to stretch the stomach. You're going to increase the gastric acid, which is going to do what? Decrease the pH, yes, right? Increasing the acid, decrease the pH. And this is going to be also kicked in by the presence of undigested products in the stomach. You're going to release gastrin, right? We talked about this a moment ago. That is going to increase the muscle contractions of the stomach and set up that churning of the stomach. It's also going to increase the release of acid from the parietal cells and the secretions from the chief cells. 3 or 4 hours of churning and churning and churning. So here it is. You think about it. That's cephalic phase. You're sending signals down your vagus nerve, preparing your stomach, telling it to make more acid and substances in preparation of everything. Then you jump into the gastric phase. The 3rd phase is the intestinal phase. By now, the chyme has now entered into the duodenum. You've also got the distension of the duodenum. This is going to inhibit your gastrin production. It's going to decrease mobility. Why would that be? Why would you decrease motility? Once I've got food in my gut, I want to slow down motility so that I have time for adequate absorption. So I do want to control this. So it's not all about zooming it up faster. This actually helps to slow everything down. This is going to stimulate the contraction of the pyloric sphincter and decrease chyme released into the duodenum, because you're going to want it to sit there for a while. You want it to move from the duodenum into the jejunum and have plenty of time for absorption of the nutrients. So here's that intestinal phase. Right, food is dropped into my duodenum. If it was sugar, what's going to be upregulated? Go back and look. What is it? GIP. If it was fatty, CCK, right? And so you see those hormones, right, that are being released as a result of food substances coming into the intestine, and all that feeds back and talks to your stomach as well. There are also reflexes here as you stretch the stomach wall. I already told you the stomach has some plasticity. As you stretch it, it does not automatically squeeze back. But as you are distending the stomach, you are going to increase your gastroenteric reflex. Basically, this is going to increase motility and increase secretions along the intestine and trigger the ileocecal valve. So the ileocecal valve is what? The one that goes from the ileum into the cecum. It's the last control. It's the last part of the small intestine going into the large intestine. Basically, so what does that mean? If you are stretching the stomach, you got to make room for what's coming, right? So how are you going to make room for what's coming? Open up the ileocecal valve. Really? Does that make sense? So the food's coming. You got to make room in the intestines for it. So we're going to open up the ileocecal valve and allow what's already been digested to push into the large intestine for the compaction and creation of the feces. So here we go. In this picture, as you stretch on the stomach and have your lunch, that distension is going to cause an increase in motility along the entire intestine, kind of pushing things through, right? The next meal's coming, fourth meal, right? You've been to Taco Bell at midnight, so you got to make room. So push that stuff through, and then also open up the gastro-iliac reflex. It all makes lovely sense, doesn't it? The large intestine, also called the colon or the large bowel. This is not as long as the small intestine. It's divided up into 4 regions, as we'll see, and its functions really are to reabsorb what little bit of water is in the chyme, to absorb a few more vitamins, mostly through bacterial action. So you depend upon the bacteria in your gut to actually absorb and create some of your vitamins. And finally, store that fecal matter until it is defecated. 3 regions here: cecum, colon, rectum. And I would argue the colon is then divided into 4 parts, right? What are the 4 colonic parts? Ascending, transverse, descending, sigmoidal. Right? So we know even more than this is telling us up front. The cecum is a little pouch at the very end as you leave through the ileocecal valve, and this is where your compaction of feces begins. Little cecum. Now, what is hanging off the cecum? We know this. It's the appendix. What does the appendix do? Nothing, although, right? It has it's lymphatic. It has a large number of lymphoid nodules in it. It does function as a lymphoid organ. Can we do without it? Absolutely. But it does have some function. It's not a complete nothing. We also think that it stores good bacteria. So in that little appendix, there's also the storage of some critically important bacteria for the digestive process. What is the problem with appendicitis? If it's inflamed and then it bursts, what's the real issue? The bacteria of the gut, right, are going to now be everywhere. Those bacteria would have been controlled and contained within the gut tube, and now they're everywhere. And now your body goes into this systemic fight against bacteria. So what cells are going to be recruited in large number? Neutrophils, right? Now, and all those neutrophils and cells, and then all the bacteria also can overall cause all kinds of infection problems. So the problem with appendicitis is not the appendix itself as being the problem. It's that you've released bacteria into the gut and you have a massive infection that the body has to deal with. So here's the colon again, the 4 parts of it. We know: ascending, transverse, descending, sigmoidal. And the ascending and the descending colon are retroperitoneal, meaning what? They don't have peritoneum around it. They don't have a serous membrane around them, and they are directly attached to the abdominal wall by the mesocolon. Remember the mesocolon we talked about at the very beginning, the mesenteric structures that attach the colon to the wall. The ascending wall bends sharply at what's called the right. Usually you'll see the word hepatic flexure. I like the second word better. That's what you'll see more often. The right colic or hepatic flexure. So basically, the ascending colon is going to turn at the liver. The transverse colon is then going to continue, and it's going to have a sharp turn at the spleen, right, at the splenic flexure. Then the descending colon is going to travel down and then go into a S-curved sigmoid colon, and that's going to eventually empty into the rectum. The rectum is the last 6 or so inches of the digestive tract. It is relatively straight, therefore the term rectum. It is expandable for the storage of feces. It's also here in the rectum that the feces are going to be pushing on the anal sphincters. There are 2 anal sphincters, recall. The internal sphincter is your urge to go, it's your smooth muscle, and then the secondary sphincter is voluntary. Along the intestine, there are some appendices. There are some little sacs of fat that are sitting on the serosa of the colon. And the Tenia coli. This is a band of smooth muscle. It looks, on the models, it almost looks like I'm going blank now, but it looks like a cinch line. Something you're going to pull the house drawer, those sections of the colon, and it runs along, and it's just the Tenia coli. It's going to be basically the outer layer of muscle in part around the intestine. The haustra, each of those little pouches, sections of the intestine, the little houses are each called a haustrum, and they are also created, these little sections are created by the accordion contractions of the Tenia coli along the way. They're also going to be able to expand. So the colon can expand and retract along the way. So looking at this picture, each of these little sections is a haustrum. The Tenia coli is this line that we see traveling along. These little appendices, here they're just called omental appendices. Other books will call them epiploic appendages. Again, just little fat. I don't know of any purpose of them, but these little fatty, appendages, right? Little fatty sections that are traveling along the colon. They're just there. They just are. Notice that there's a lot of blood vessels, of course, going to the intestine, and we do not see any nerves here, but there are also a large number of nerves traveling to the intestines as well. Up here, right, this flexure, this sharp turn, that's your hepatic flexure. The liver's over on that side. Over on this side, this sharp turn is the splenic flexure. This one is called the sigmoidal flexure as you go into the S curve, as you go into the sigmoidal colon. Now, there are very large, powerful peristaltic contractions that occur a few times every day. As you distend your stomach and the duodenum, this is going to push products toward your transverse colon and toward the end of the large intestine. So those big movements are really going to be pushing the feces, or creating the feces, toward the end of the colon. Now, if I were looking under the microscope at the colon, what I would see is that there are no villi, meaning there's no what? Big folds. We're not worried so much about increasing surface area here. Our job basically is to lubricate the feces and get them more compact. There are mucus cells along the way. The mucosa does not produce enzymes, aka there's no brush border. Right, the small intestine had the brush border, the microvilli with the enzymes. The large intestine, there are no enzymes being produced by the mucosa. So it's much, much smoother in appearance. The lining, simple columnar. There are glands that are going to be creating mucus that are going to keep this nice and moist. And still a muscularis made up of 2 layers. That hasn't changed. The last part, as you leave the rectum, you enter into the anal canal, and there are 2 sphincters here. We change also from columnar, simple columnar, to stratified, squamous, non-keratinized, right? So where did we have that before? In the oral cavity esophagus, right? We started out with stratified, squamous, non-keratinized, and now we finish with that as well. There's also a series of veins here, and if there's increased pressure on these veins, they can extend in what is commonly called hemorrhoids. Right? So those rectal veins can distend and even distend out of the anus, and that happens during the straining of defecation if you're constipated, as well as oftentimes during pregnancy. Those suppositories, right, the hemorrhoid suppositories are doing what? Little pain relief, but also helping to dilate, right, and close down those distended veins. There's two sphincters here at the rectum. There's the internal sphincter, as I've already mentioned, that's the smooth muscle that is involuntary. The outer is the external anal sphincter. It is voluntary muscle. And then finally, you go from non-keratinized to keratinized as you travel out of the anus, and now you're basically along the skin. You would now transfer to keratinized, right? Because everything on the outside of your body is keratinized. So if you had a nice little histological section of the rectum and the anus, you would see those distinct changes in the histological continuum, right? From simple columnar to stratified squamous, non-keratinized, to stratified squamous, keratinized as it exited the body. So again, that rectum, straight region, last 6 inches or so, and then these sphincters you can see are sort of not really that distinct, but you can see two different layers of muscle there, internal and external. The job of the large intestine is only to absorb about 10% of the nutrients. It is really all about water, but also vitamin absorption. So biotin, vitamin K, vitamin B5, these are actually produced by bacteria in your colon. You've got to have bacteria in your colon to make these vitamins. This is also where you're going to be reabsorbing water, whatever's left, and about 1,500 mils of material will enter the colon. A liter of it will be reabsorbed through osmosis, and that leaves about 200 mils of feces that are ejected. So with those 200 mils of feces ejected each day, 75% of that is still water. The rest of it is the inorganic and indigestible products that you could not digest. The rest of it is also epithelial cells and bacteria. So there's a large number of epithelial cells that you're defecating along with a lot of bacteria, and those bacterial compounds are producing to the odor of feces, so the ammonia, other nitrogen-containing compounds, as well as the lovely hydrogen sulfide. The stinky egg stuff, the farts, that's the rotten egg odor. And that's all made by the bacteria of your gut. So in the large intestine, we're going to be absorbing vitamin K, B5, biotin. Those bacteria are largely in your gut and helping you do that. And you're reabsorbing a lot of water as well. So we can't live without our bacteria, right? They are important for us, not only as a protective layer on our surface of our body, but also lining the digestive system. As you begin to defecate, there's a reflex here as well as the rectum is distended through the packing of the feces. Then there's going to be a triggering of stretch receptors in the wall of the rectum. And this is going to involve two positive feedback loops. Okay, what do we know about positive loops? They keep getting bigger and bigger and bigger, right? Moving further and further away. And so the long reflex is coordinated with the parasympathetic system. This is going to be pushing by mass action the feces toward the rectum. And then there's a short reflex. The muscles are affected by the Mesenteric Plexus. And this is going to kick in the colon and rectum. Together, it's going to cause a relaxation of the internal sphincter, move things further along, and then eventually cause defecation only with, hopefully, the relaxation, the voluntary relaxation of your external sphincter. This control of this process does go back to, reflexes are all involved with your brainstem and your spinal cord. So there is, if you had brain damage issues here or spinal cord damage, you could have loss of control over this process, as you can imagine.
The same thing as like with
You can't
But it
hold,
will
you
be coming
right,
down to-
right. You cannot hold it forever, right? It will happen, and hopefully when you are sitting over a bowl of water, right? So it will happen, right? I mean, this is something just like breathing, like Stacey said, you can't control it. At some point, the body will take over and say, hey, dummy, we got to take care of business, and we'll do this for you. Okay, moving on. The last little bit are the accessory organs. So I've finished up all the truly digestive organs. We've gone from mouth to anus. We've talked about the reflexes of swallowing, the hormones of the digestive system, the control of movement through the intestines, and now we're moving into the accessory organs. Here, I want to spend a good few minutes talking about the salivary glands, but especially about the liver. Okay, that's really what I want us to focus on. Love your liver. So 18, just naming the accessory organs, this is not my concern. Salivary glands, parotid, submandibular, sublingual, you've heard of those before. The gallbladder is storing the bile made by the liver. The pancreas is releasing hormones that we've talked about before, but also releasing the bicarb and helping with the digestion of proteins. And then the liver has tons and tons and tons of things that it does. We talked about the liver in the blood section, right? The liver makes the blood proteins, but the liver does so many, many other things. So that's where I want to focus our attention over the next 10 minutes or so, is on the liver. So this first part is just about those accessory organs and how they contribute to the process. The salivary glands, they are producing saliva. They are controlled by cranial nerves 7, 9, and 10. Do you agree that taste buds are 7, 9, and 10? 7 was the first, or the 2/3 front of your tongue. 9 was the back 1/3 of your tongue. And there are a few little taste buds way back on your epiglottis, and that information is picked up by your vagus nerve. So you will see vagus included in taste, just to be complete. And then your salivary, the receptors of this are being picked up by the trigeminal. Remember, the trigeminal is the one that has the 3 regions, ophthalmic, maxillary, and mandibular. And this is all about parasympathetic. So if you think about a meal, you're going to start making more saliva under parasympathetic control. Sublinguals, under the tongue, makes sense. Not big players. A small percentage of your saliva is coming from them, mostly lubrication. Those submandibulars, they're 70%. They are your major salivary secretors. They are releasing enzymes called amylase. Amylase is a sugar starch breaking down enzyme. So sugar starts being broken down in your mouth. That's not true of proteins. Or if you're chewing on a piece of chicken and you get that piece of chicken stuck between your teeth, in the morning, it's still there. Right? So you're not breaking down proteins in your mouth, but you are breaking down sugars. So if you take a saltine cracker and you take, or a piece of bread, and you let it sit in your mouth, it becomes kind of sweet, doesn't it? That their starches are being broken down in your mouth into simple sugars, so you'll feel that sweetness approaches because sugars are first broken down in your oral cavity. You also have here some antibodies. You have antibodies produced directly into your saliva. These antibodies, like any antibodies, are trying to fight off foreign antigens. And these IgAs, these type of antibodies, are released into your oral cavity to help fight off bacteria and pathogens that would enter into your oral cavity. You're also making, I didn't write it down here on words, but you're also making growth factors here that are helping to repair your oral cavity. Should you get burned or cut your tongue, it grows back really, really fast.
The thing where dogs lick their wounds.
Dog lick their wounds. Definitely lick your wounds, guys. Spit on them, spit on each other, lick your wounds. The saliva has wonderful things in that, very regenerative, very good for epithelial cells. Right, so great stuff. The parotid glands are the biggest glands. They're secreting about 1/4 of the fluid. Similar in what they release. Now, where your dental hygienist, when you go have your teeth cleaned, where the dental hygienist is working the hardest is right behind your bottom front teeth and back around your molars. And part of that is because that's where these ducts are for the release of your saliva. And these salivary glands release proteins, and those proteins gunk up and create that tartar and calculus, which needs to be taken off. So the problem areas in your mouth are right next to where your salivary glands release their products into your mouth. So there we can see them, right? The submandibular, the sublingual, and the parotid glands. Beautiful, beautiful things. Okay, now, do you agree that when you are thinking about supper, and you're in that cephalic phase of digestion, that you start making saliva, and that saliva is more watery, right? As you anticipate a meal, your saliva is more serous, more fluid-like. But if I then asked you to stand up in front of a crowd and give a speech, all of a sudden you go dry mouth. When the bear walks in the room, you don't have watery saliva, your mouth becomes very dry, very sticky. Why? That's very mucousy. So your salivary glands are mixed. They can release watery, serous secretions, or they can release thick mucus secretions. Why do you think your saliva, your salivary glands, is more mucousy, thicker when the bear is chasing you? Protection of. What are you doing more rapidly when the bear is chasing you? Breathing. And can you imagine how quickly the airway would, quote, dry out when you're exercising or breathing quickly? So that mucus, that thickness that's secreted by your salivary glands is actually to protect your airway from that rapid airflow that's going up and down. Whereas when you're about to eat, right, the same glands secrete more of a watery, let's get down to business, let's have a meal, let's digest the food. So we consider the salivary glands mixed because of that. There are also antibacterial enzymes secreted directly into your saliva as well. I will not ask you to recognize histology, okay? But these would be exocrine glands, and they're going to look like, they're going to have secretory vesicles that release the products. Saliva, 1 to 1.5 liters a day. Most of it, by far, is water. It's there to flush your oral cavity. It's there to keep the bacteria are in your mouth. Those bacteria are constantly making acid. And if the saliva is not flowing adequately, what happens to your enamel? People with low salivary output, dry mouth syndrome, right, have a bigger issue with caries, with cavities, because the bacteria are able to make the acid, and we know what acid does to limestone. We know what acid does to bone. Acid breaks it down. So the enamel gets broken down. Once your enamel's gone, folks, it's gone. It's like cartilage. It doesn't get regenerated. So enamel, you're born with it. There's a layer of it there, and that's it, right? And once that enamel's gone, it's gone. So it's very important to protect it, obviously. Okay, now done the liver. 7 minutes, let's talk about the liver. I'll probably pick up with the rest of this. This is your largest visceral internal organ. It is wrapped by a tough layer, sort of like the kidneys are. It is covered by a visceral peritoneum. So that means it's what? It's in the peritoneal cavity. It is composed of 4 lobes, right, left, caudate, and quadrate. Caudate means sort of what? Toward the tail. As you'll see, the shape of it, right and left, pretty straightforward. So where is the liver? If we look at this cross-section, you can see this big old sucker, right? This is all liver here. And right here, remember back to the beginning, the falciform ligament is connecting the liver to the wall. You've also got the sternum right here, right, up in the front, and little spleen on the other side. So you've got your stomach with liver on one side and the spleen basically much smaller on the other side. Left lobe, right lobe. Those 2 lobes are connected or separated by the falciform ligament. Okay. There is an area called here the bare area. It's an area where there is no peritoneum, and it's where the liver comes in direct contact with the diaphragm. The round ligament is a thickening of the falciform ligament, and you'll see that it marks the path of where the fetal umbilical vein once was. The caudate nucleus is, quote, more the tail. And then the quadrate lobe, I said nucleus a moment ago, caudate lobe, and then the quadrate lobe is between the left lobe and the gallbladder. It's the smallest of them. What else do we see here? There's the gallbladder. The gallbladder is the storage place of bile. Bile is made constantly by the liver, but is stored in the gallbladder. And when what comes into the gut, fat, the duodenum makes CCK. CCK tells the gallbladder to squeeze and release that bile into the duodenum for the mixing. There's also the Porta Hepatis. This is, quote, the doorway to the liver. This is an area with the blood vessels that are entering the liver, and we'll see how this all goes together with the common bile duct. So let's take a look at the lobes of the liver. Left, right, easy enough. They're very obvious from the anterior surface. What's separating them? Falciform ligament, right? Falciform ligament separating left and right on the front side. On the back side, okay, on the back side, this little tiny area between the gallbladder, that is the quadrate lobe, and then the caudate lobe is this other portion. So really those are the two on the back side. The bare area is the area that's making direct connection with the diaphragm, so there's nothing there, sort of, it's the bare area. The Porta Hepatis, this is the area where you see the common bile duct, the hepatic artery, and the hepatic vein kind of going in. You can also think of that sort of like a hilum, right? Remember the kidney has a hilum where everything goes in and out? Sort of like that. That's where most things are going in and out of the kidney at the Porta Hepatis. Think of the port, right, going in and out, where everything's going in and out of the liver. So that's the anatomy of the liver, these lobes. Now, I want to introduce this, and then I'll pick up with this little bit at the end as we talk about what the liver does. The liver does lots of things, right? It makes the proteins for your blood. Here we're talking about how it is going to filter your blood. It is going to detoxify and filter what first enters into your bloodstream. The unit that's going to do this filtering is called the lobule. So there are lobes made up of many little lobules. There are over 100,000 liver lobules. Each one's about 1 millimeter in diameter, and they are separated by a septum, just like your nasal septum, something that separates all of these little all these little lobules. Each of these little lobules is hexagonal in shape, 6 little sides to it. So let's take a look at what this means. Now what we're going to see is that in the liver, there's a portal area, specifically a portal triad. 3 things in the portal area. And what you're going to see is that there's a branch of the hepatic portal vein. What is that doing? Hepatic portal vein was bringing nutrients in that were just absorbed into your gut, and they're bringing them toward the liver. There's also a branch of the hepatic artery. The hepatic artery is bringing blood to the liver. And then there's also a little bit of the bile duct here. And we're going to see that the arteries and the veins and the bile are all going to be intersecting here in these lobules. The cells that make up the liver are called hepatocytes. Hepatocytes, liver cells. And we're going to see them make these lobules, these plates. We're going to call them plates. And there will be some short microvilli here to increase the surface area. What is the sinusoid? What's the sinus? Space. So we're going to see these liver sinusoids, these spaces created in these lobules. Here there are very delicate blood vessels that lack a basement membrane, large fenestrations. Another way of saying these vessels is that these are discontinuous capillaries. Another name for those would be discontinuous or sinusoidal, right? So capillaries come in 3 basic types: continuous, fenestrated with little openings, and discontinuous with big openings. That's what we have here. Very large spaces that allow all of this blood flow to come in. So let me introduce this hepatocyte, and then we're done. My last slide. We'll pick up here. What I'm looking at is this hexagonal structure. Do you see the 6 sides? Okay. at each of those 6 points of the hepatis, of the hexagon, you'll see the triad. Here's a triad, here's a triad, here's a triad, here's a triad, there's a triad, there's a triad. That triad is made up of a portion of the hepatic portal vein, a portion of the hepatic artery, red, and a portion of bile duct, green. So that's why you're seeing red, blue, and green. And then in the very center, there's a central canal, and that central canal is going to be receiving what comes in here. So we're going to be talking about this on Thursday. We'll pick up right here. I'll kind of back up a little bit, walk you into what's going on in the microstructures of the liver. That really only leaves us with a couple of pages. There's not a lot left on the digestive system. Please, I know some of you have the lab exam still coming up, but if you've already had the lab exam, don't feel, like you can't go ahead and start looking at the digestive mastering assignment and the lymphatic mastering assignment. The more difficult portion of this next exam will probably be the respiratory. There's just a little bit more involved with respiratory. So this isn't so bad. Lymphatic's not so bad. Respiratory will take a little bit of more time. So leave yourself some time to let that sink in on Thursday and next Tuesday.